This video is going to show how to make a smear from a solid culture and then gram stain it. So what is a gram stain? It is a staining method developed by the Danish bacteriologist Hans Christian Gram, who realized it separated the two largest classes of bacteria. The staining procedure distinguishes gram-positive cells from gram-negative cells. It also allows you to see size, shape, morphology, and whether or not you have a pure sample. So how do you gram stain? There are four components: crystal violet, Gram's iodide solution, alcohol, and safranin. So what do each of these do? Crystal violet acts as a primary stain, Gram's iodide as a mordant, which sets the stain, alcohol acts as a decoloring agent, and safranin as a counterstain. Before you stain, you must make a smear on at least 2 slides, not just one in case you mess up. You draw a circle on the bottom of the slide, label it, and then flip it over. You have your incubated culture in colonies, so what next? Take your loop and flame it for a few seconds to sterilize it, then cool it for 20 to 30 seconds. If you don't cool it, you'll kill all your bacteria. Next, take some of your sample from a colony that appears pure. On each of your slides, add a drop of water and rub the loop back and forth to spread the bacteria across your circle. Then, let the slides air dry. Once dry, heat fix the slides by swiping them back and forth 3 times across the flame. This secures the bacteria to the slide and prevents distortion. Place your slides on the boat and prepare to stain. First, take the crystal violet and cover the slide for 30 seconds. Then rinse off with water. Next, add Gram's iodide for 30 to 45 seconds. Do not rinse this off with water, but have water nearby. Instead, use a few drops of alcohol, just until there is no longer color running off the slide. This acts to decolorize. Then, quickly rinse the alcohol off with water. Finally, cover the slide with safranin for 30 to 40 seconds. Rinse the safranin off with water and then pat dry with bibulous paper. To do this, you place the slide under one sheet and then cover it with the sheet and then blot dry. Now you have 2 beautiful gram stains. So what do you do with these? You analyze the slides under the microscope and determine what kind of bacteria was in the colony that you chose. Gram-positive bacteria will be purple, in this instance cocci, and gram-negative will be pink, in this case rods. So why do these cells turn different colors? Gram-positive and gram-negative cells differ in many components. Gram-positive cells have a plasma membrane and a thick cell wall, where gram-negative cells have a plasma membrane, thin cell wall, and outer membrane. When talking about a gram stain, the most important difference is the width of the peptidoglycan layer, or the cell wall. These are made up by the NAM and NAG units. Because the gram-positive cell wall is thick, when the alcohol is added, it dehydrates the cell wall, retaining the purple color inside. But the gram-negative cell wall is thin and is degraded by the alcohol. So when the alcohol is added, it simply gets decolorized. When the safranin is added, it turns the gram-negative cells pink because they no longer have any color in them. But it does not show up on the gram-positive cells because the purple pigment is much darker and covers up any of the pink that would show. Gram staining is the first step to determining if you have a pure culture and what that culture is. Happy staining!